All right, that was a really great talk, Mark. I'm Anna Malofsky, and nice to see everybody. And it is my pleasure to introduce Richard Jin. So Richard was an undergrad at UCLA in biochemistry and did his MD PhD at SUNY Buffalo, where he worked with Elizabeth Wolfert. As you'll hear from him, and I'll keep it brief, he was an immunologist by training and didn't study the brain at all. As an MD PhD, he got fellowships from AAI, the American Association of Immunology. He got the Dean's Award for Outstanding Dissertation Research, and then he came here to UCSF for his research track residency. And he has joined my lab, as you'll hear. He's been full-time in the lab for less than a year so far, so that's a time period where I was still trying to figure out which way was up in my postdoc. And as you'll see, he's done really incredible work already in establishing this new model that he'll tell you about. And I'm so fortunate to have Richard in the lab because of his very unique training background and his expertise and his passion for child psychiatry, which I think has really motivated the work that he'll be telling you about today and has let us take some really, really basic science findings into some directions that I think will be really clinically exciting.
Thanks, Anna. Let me adjust this microphone. So as Anna mentioned, I'm Richard. I'm one of the current PGY4 research residents here. And many of you may already know me, but if you don't, you might recognize my name from the multiple notes or referrals I've probably sent you guys throughout the years. But what you might not know about me and what Anna alluded to is that I'm actually an immunologist by training in my PhD, and I've spent a lot of my early research career being fascinated by how the immune system defends against foreign pathogens and how these immune responses are regulated so that we don't cause collateral damage in the peripheral tissues. The specialized immune cells that I work on in the body are called macrophages. And but when I returned back to medical school, I found that I was similarly captivated by a very different field, and as you might imagine, that was psychiatry, and in particular, child and adolescent mental health. And so when I came to UCSF, I was excited to start working with Dr. Malofsky, who studies the macrophages of the brain, the microglia, and in particular in brain development. And I've been full-time in our lab now for about 7 to 8 months now. And so today I'm going to talk to you about a project that I've been working on to better understand how the immune system and in particular microglia respond to viral infections and its consequences on the developing brain and behavior. And to me, this is exciting because we're in an era of science, especially in immunology and neuroscience, where we can really start to better define the cellular basis of how early life illnesses and immune activation impact long-term trajectories, and then take actionable steps in the clinic. So I suspect the concept of illness and getting sick is not a foreign one. And in fact, if anything, after COVID, I think we might be intimately familiar with it. And this is a picture of my oldest, Addie, and she gave me permission to give this, to show her picture here. She was like, "Oh, I hope that you find this, your work friends find this funny." But this was a picture that was taken of her when she was sick, and she had caught one of those daycare mutated stomach viruses, so a particularly strong virus. And you might not be able to tell beyond kind of her puffy eyes, but she had thrown up a couple of times before this picture, and she was being a good sport about this. And she knew she couldn't go to daycare, so she took that to mean that we were going to the zoo instead. So we did. And this is a picture of her a couple of hours later at the zoo. And I think we've all been here before, actually. And inherently, because of this experience, we know that infections affect our behaviors. When we're sick, we want to huddle under blankets. We want to withdraw from the world until we start to feel better. And sometimes we're a little bit more irritable. Our thinking becomes foggier, sometimes slower. We call this sickness behaviors. And while we consider most viral infections to be monophasic with a quick onset and then relatively rapid resolution of symptoms so that we can quickly get back to work. What COVID actually has highlighted for us is that even after infection has thought to have been cleared, we can continue to have lingering cognitive and mental health challenges. And so if you take long COVID as an example, this is a recent 2-year follow-up study of patients who had symptoms associated with COVID and have extracted out some of the more relevant neuropsychiatric symptoms. The intensity of brown reflects an increased relative risk, and as you move from left to right, that's the time course after infection. And so what I hope this graph highlights is that there are certain symptoms, including fatigue, memory problems, sleep disorders, and even suicidal ideation, that at this 2-year mark, there still is persistence of in a proportion of patients who had COVID. And then there are other symptoms like depressive disorders and generalized anxiety that also resolve, but resolve much slower than we would maybe expect from other viral symptoms that are more somatic in nature. And so this is not unique to COVID. In fact, we call this post-viral syndromes. And in my own specialty panel here at UCSF, I see patients with new and chronic psychiatric symptoms resulting from prior infections in autoimmune diseases. And so we know infections and inflammation have a long-term impact on behavior. And so that data was from adults, but infants and kids are particularly vulnerable to viral infections. This is in part because their immune systems are immature, and just like the developing brain, they're learning too. They're learning good from bad, they're learning how to efficiently fight off infections, all the while they're retaining memory for the next round of infections. And brain development is a highly organized process, and presumably even transient insults can have lasting impacts. And while most viral illnesses are self-limited, what epidemiologic studies have shown us is that exposure to both infections and increased immune activation in early life increases the risk for developing mental health illnesses. And so there have been multiple epidemiologic studies, but I'm going to highlight one of these here, over here on the left, or I guess that's your right. This is a more recent Danish registry study of about 1 million children, and they wanted to investigate the association between all treatments for infection since birth and subsequent risk of development of all treated mental health disorders during childhood and adolescence. And so to do this was mostly a study looking at ICD codes for either a hospitalization for infection or a prescription for an outpatient prescription for an antimicrobial. The hospitalization for infection they were using as a surrogate for more serious infection, and then prescriptions for outpatient antimicrobials was used as a surrogate for more mild infections. And then they interrogated ICD-10, 8, 9, and 10 codes for mental health disorder diagnoses or having received a psychotropic medication prescription. And so what they did find was that there were increased risks in children who had been hospitalized for infection and then subsequent diagnosis of a mental health disorder. And then that was also true of more mild infections, or what they used as kind of that readout for more mild infections, so the prescriptions for antimicrobials. I've also provided on the bottom this Carlson paper, another epidemiologic study, a Swedish cohort study or Swedish registry study that looked at the impact of any specialty care received for infections and then the later development of autism or intellectual disability. And while I mentioned that there are multiple epidemiologic studies, I think they all kind of hint at the same thing, which is that there is potentially a link between infection and mental illness, including neurodevelopmental disorders. But how this happens is a much trickier question, and a question that's harder to study in humans, especially in children. And so the question that, the fundamental question that I'm asking is, how does early like viral infection affect brain development and behavior? To answer this question, I want to introduce the major immune cell type in the brain, and I alluded to this before, but this is the microglia. They make up about 10% of all cells in the healthy brain. And at rest, they have this highly branched appearance with multiple primary and secondary processes. And as you might expect, being an immune cell, they are very efficient and help in fighting off pathogens. And they produce many immune molecules that either directly kill virus or signal to other cells to kill virus or to kill pathogens. They also are professional eaters, so they eat a lot of infected cells and pathogens as well. But what we've come to realize as a field is that immune cells also play multiple physiologic roles, and microglia in particular are very important for brain development, performing multiple functions. But microglia don't do all of these functions all at once. In fact, microglia are oftentimes very much specialized to do a specific task over another. And so what our lab has recently discovered is a role for a new subtype of microglia that take on an antiviral-like state. And so they turn on a lot of the machinery that typically is used in viral infections, but during healthy brain development where there is no infection present. And this is work that was spearheaded by Leah and Caroline in our lab. And it's now in press. And if you were here last year, you saw Caroline describe some of this work. And so the dots on this graph represent an aggregate of individual microglia that were isolated from the developing brain, either at rest or in a model of cortical remodeling. And each cell was sequenced and computationally assigned clusters according to similar gene expression. And so the microglia that I'm alluding to are here in cluster 8. And one of their hallmarks is this expression of an interferon-stimulated gene, IFIDM3. And so this is a product that is involved in antiviral responses, but we mostly use it, at least for the purposes of this talk, to mark our antiviral-like microglia, or our microglia that have assumed this antiviral-like state. And globally, they actually represent a very small proportion of microglia in the brain, but in a model of cortical remodeling, they actually expand 20-fold. And so here on the right, I have a picture of what they're doing, right? So we've identified them. What are they doing? I've labeled microglia, or we've labeled microglia here in green, and then they're also expressing this IFIDM3 or this antiviral response. So these are these antiviral-like microglia, and you can, when you look at this picture, you can see that they're not this kind of highly branched microglia that we typically saw at rest. In fact, the morphology has shifted such that you can see this microglia start to reach out its process on the top towards a cell. And so what this microglia is doing is starting to reach around and engulf this whole cell. And then on the bottom, it's not just one, but it's two. So these microglia are highly phagocytic, so they're super eaters in this context. And what we know is that when we inhibit the ability of microglia to enter this antiviral-like state, we have an excess of cortical excitatory neurons and also have an increased tactile hypersensitivity. And so this is an increased sensitivity to touch. So these microglia are doing a very important role in development when circuit remodeling is taking place. But what happens when you have too much of a good thing? And I mentioned this machinery is shared with antiviral responses. So what happens during an infection? I'm hypothesizing that in viral infection, there's an overexpansion of these super eaters, and that leads to excessive damage in the brain and long-term behavioral consequences. So to test this hypothesis, I wanted to first create an early life viral infection model that was transient, that allows us to study the brain both during infection and after, so that we can interrogate things like what is the immune response during infection, and then what are the consequences of this immune response or infection. I collaborated with Raul and Dino in virology, who's an expert in enteroviruses. And I selected enteroviruses because as a family, they actually are the most common infection of childhood. They're a single-stranded RNA virus with 13 species. The most commonly known is actually polio, but we use Coxsackie B3. And I selected Coxsackie B3 because one, it infects through the gut and the nose and has a mild transient course in kids. And when they do have symptoms, they typically manifest as upper respiratory infection like symptoms or hand, foot, and mouth. But on a population level, it isn't neuropathic, so it doesn't typically cause overt clinical neurologic symptoms. And so what I did was I infected 2 to 3 day old mice. So these are very small pups. And I tested various titers of virus by infecting them through the gut to mimic a more naturalistic infection. And it's common to use weights as a surrogate for illness and recovery. And what I'm showing you here is actually a titer that I used that struck that balance between having the mice get sick but not dying, so that we can study the mouse and infection throughout all phases. What you can see here in black is the weights of the uninfected mice, and in red are the infected mice. And what this graph depicts is a transient weight loss between 5 to 10 days, but the infected mice rapidly regain their weight to their uninfected counterparts. And in fact, if we do some clinical scoring on these mice, what we see is that they do get sick around days 8 to around day 13 or so. And then they resolve in kind of these more clinical symptoms of illness. And so this is the model that we have. But most importantly, all the mice survive, so we can study the consequences of infection. So now that we have our model, we can start asking what is happening in the developing brain during infection, and more specifically, do we have induction of antiviral microglia? So when I look in the brains of these mice 7 days after infection, and so this is during a period of weight loss, but right before they start to look clinically unwell, I can see clear foci of where virus has entered the brain. And so the virus here is labeled in orange. And this is surprising because Coxsackie is not thought to actually enter the brain or have tropism for the brain. And both clinically as well as what we've seen in our model, there is an overt neurologic symptoms. And I see that in these corresponding areas, what I've labeled here in magenta is this interferon response or this antiviral response. And so the immune system is responding to virus in these pockets. And then in green here are the microglia. They also are enriched in these areas where you have these foci of infection. And so I looked at earlier time points to see what the immune response was like, and the kinetics. And what we see is actually the interferon response comes on pretty early, even before the virus is visibly detectable by microscopy in the parenchyma of the brain, and then both of which are happen before clinical symptoms. And so interestingly, there are hotspots for infection. And so a couple of these you might be able to recognize here in the picture on the left. But when I plot them across 4 biological replicates, each of these replicates are in unique colors here in this graphical representation. What we see is that these hot spots broadly tend to enrich in the cortex, in the thalamus, and in the midbrain. And so what I hope to eventually answer is if there's something unique about these areas that makes them more permissive to infection, and ultimately, are these regions important for the behavioral outcomes I'm about to show you. Also, I don't want to miss this, but the brainstem looks like it's pretty inflamed as well, but that's probably something that we will tackle a little bit later on. So when we zoom in on one of these hotspots, what does it look like? In green, I've labeled microglia again, but I've traced 2 microglia here, one in red and one in yellow. And at first glance, what we see in this morphology is it doesn't look like these microglia that were at rest with kind of these branchy processes. And so they're actively doing something different. I've labeled the nucleus of one of these microglia here, and around the nucleus, we have these antiviral proteins. So these are antiviral microglia. We also have these structures which are kind of circular in nature. So these are actually phagocytic cups. This is when the microglia is circling around actually multiple cells in this picture, some of which are clearly virally infected in white, and others are not clearly virally infected, but in either case, they're engulfing multiple cells in this picture. And it's not just one microglia, but there are multiple microglia that are doing this. And if you compare these true antiviral microglia against the antiviral-like microglia that we saw during in our cortical remodeling and in healthy brain development, they do share a lot of similarities, both in morphology and the fact that they are engulfing cells. But then 30 days post-infection, actually, there are no more infectious nitises, and there's no clear evidence of an increased interferon response or antiviral response. And so globally, things have returned back to prior. So returning to kind of this model and building on it, what we see is that early during infection, there's a broad and early antiviral response marked by induction of these highly phagocytic antiviral microglia, and that resolves when we look 30 days post-infection. So what are the behavioral consequences of viral infection when we look 30 days after infection or beyond? And so these are my kids again. And I wanted to show you these pictures because touch is such a big factor of our behavior and our development. And I emphasize this because the set of behavioral experiments I'm about to show you are related to tactile readouts. It's also a big component of many neurodevelopmental disorders. And in many of these neurodevelopmental disorders like autism, there's tactile sensory processing that's impacted and manifests behaviorally. So on one hand, we have this tactile hypersensitivity, which can manifest as irritability and agitation or a desire to withdraw when overstimulated. And on the other hand, we have tactile hyposensitivity, which can manifest as kind of that same irritability, but can be seen when sensory requirements are not met. And this can manifest as sensory seeking behaviors, wanting to use weighted blankets, increased touch, and these two phenomena are not mutually exclusive. So the first assay that I'm going to show you is actually a tactile hypersensitivity behavioral paradigm called the whisker nuisance test. This is a pretty simple assay in which we just tickle the whiskers of mice and see how annoyed they get by this assay. Mice are largely just all whiskers, and so you can imagine they can get pretty annoyed. And so we actually score these mice on a couple of parameters here that I'm going to show, but by and large, an increased scoring reflects increased tactile hypersensitivity. And I'm going to show you this video, but before I show you this video, I will mention that typically in uninfected mice, mice are a little bit bothered by the wooden dowel initially, but then they rapidly habituate through multiple trials of the stimulation. This is from a mouse that had been infected about one month prior, so this is after the illness had resolved. And so what you can see in this video is that this mouse is constantly moving. It's kind of batting away at this stick or grabbing on it. And at the end of this video, it's actually even trying to jump out of the cage. And so it's pretty annoyed right now by the stick. And so when we plot the global scores here, we see that even 1 month after infection, there's an increased whisker nuisance score, meaning that these mice have increased tactile hypersensitivity. If we look at a different readout here, and this is interactions with the stick, both 1 month and 2 months after infection, there's this persistence of this tactile hypersensitivity. The next test I performed was one that was recently described by Catherine Dulac's lab, and in many ways the opposite of the last test. Whereas in the last test, we used an active aversive tactile stimuli. In this test, we call it it's called the gentle touch preference test. We allow mice to passively choose whether they prefer to go through a plush cloth-lined tube or to go through a hard plastic tube. So let me play this video for you. On the left are uninfected mice, and then on the right, this is 2 months after infection. And so while you're looking at this video, when we did end up, the way that we analyze this assay is we plot the number of crossings between each tube, whether they're a plastic tube or one of these really soft tubes, against each other, and then we come up with a preference index. And these mice 2 months after infection do seem to prefer this a passive soothing touch. And so in interpreting both of these behavioral tests, this would suggest that sensory processing differences are present as a post-infectious sequelae, at least in our model. So what did we learn? How does early life viral infection impact brain development and behavior? From what we've seen so far, we've seen this increase in antiviral microglia that enrich in certain areas of the brain. These are actively eating many cells and possibly indiscriminate of whether these cells are infected or not infected. And about a month after infection, when illness has resolved, there's no clear evidence of an active interferon response or antiviral response, we see that there are persistent tactile sensory processing differences and perhaps akin to what we might see in neurodevelopmental disorders. So what's next? I think we've really only reached the tip of the iceberg, especially after establishing this model. And there are many questions I think are really cool to kind of tackle now that we have a model in place. Some of these questions are, well, first we see that the microglia are actively eating many cells. And does this lead to a drastic difference in the types of neurons that are in the brain and the development of circuits that are important for behavior? The other question is, what are the other immune cells doing? Are they helpful in perpetuating this response or maintaining these antiviral microglia? And I think, there's question about whether immunologic memory, for instance, is constantly being activated, and that might be involved in long-term behaviors. I'm interested in further validating the tactile sensitivity phenotype, but also expanding to include and ask the question whether cognitive and social behaviors are also impacted by viral infection. And finally, we know that humans are much messier with genetic variation and things like this. But looking at gene environment interactions, and so does viral infection cooperate with genetic risk alleles, for instance, in autism, to increase certain behaviors or phenotypes that we might otherwise see. So many questions to continue to ask, and ultimately having a better understanding of viral pathogenesis and the immune response to infection, especially during developmentally critical windows of time, will be important for us to really start thinking about when we're thinking about developing tools or even programs for intervention. But I definitely think we're headed in the right direction here. So thank you. And these are my acknowledgements. The Malofsky Lab, Anna, who's my primary mentor, and everybody who I sit side by side with to have discussions with all day long about microglia and viral infections and brain development. Also the Andino Lab, Raul Andino's the PI, and then his previous postdoc, Ming Ye, who gave me the plasmid for Coxsackie B3, the research track here, Dr. Vogelmeyer in my career development, and also the funding for the research track, which I'm often reminded to put up here, and then the residency by and large for all of your support over the past 4 years. And I'll take any questions.
Great talk and super cool project. So I found the regional specificity of the infection super interesting, right?
Mhm.
So this is like, you have these two cortical regions and then midbrain. And I'm wondering, so first kind of why you think that is? I mean, is it probably due to the timeline of development and maybe those neurons are like in some period of postnatal development? Or if you infected an adult mouse, like would you get the same distribution, or could it be perhaps shifted to a different distribution, which might influence kind of mood and anxiety related behavior, similar to what you kind of showed with the COVID-19 graphs and stuff?
Mm-hmm. Yeah, no, those are fantastic questions. Actually, questions I wrestle with on a daily basis here, but I'll try to tackle both of them. The first of which is, I think that one of the reasons why they might be particularly susceptible may be because of brain borders and kind of the, for instance, the access to these regions from the periphery. Now, whether that's true or not, it's a little bit unclear. I think those are active hypotheses we're going after. It's not clear that this virus is actually infecting, for instance, neurons. They might be, but the specificity is a little bit less clear for those particular regions, and it's not thought to be neurotropic, or neurotropic in the sense of infecting neurons, though I have seen evidence of some of that. And then the second question about infecting adults, that's something that we're planning to do. What I can tell you though is when you infect adult mice with other viruses, some viruses also replicate these types of regions. So for instance, West Nile, Robin Klein, her lab has done some of that work. And when you look at the immunostaining, it's like, oh, wow, did you infect with Coxsackie? So there seems to be some conserved nature to this.
I know we are at time. I have at least one more question. No?
People need to leave. So actually, it's sort of more a comment. Yeah. So first, really, I think, in a really exciting area of work, and not meant as a criticism. And the reason I'm worried about this is that there's only a short period of time, but I think worthwhile, at least for the audience to have a sense that the issue around causality with regard to postnatal exposures and autism is a really hot and very fraught area. And I think cumulatively, when you look with a dispassionate eye at the epidemiological literature, that there are no validated postnatal exposures when you think about all of the potential confounds. And in fact, any that we thought that we have identified, when they've been appropriately controlled for a genetic risk using sibling-based controls, all of those postnatal things have disappeared. And it's particularly challenging because the emergence of autism is so early. And so even the temporality of these kinds of studies that suggest that hospitalization for an infection and then subsequent prescription of a psychiatric medication or subsequent diagnosis. So the reason I say that is I think it's absolutely plausible what you're describing, but it also feeds into a narrative about kind of the overall vulnerability in early life to activation of the immune system, which plays into potentially a narrative around vaccines and in early life and autism, which again has been roundly debunked. So I just say that in an audience that may not be as sophisticated as ours, that really being careful about kind of what we know about causality. And by the way, there are other examples where we know, I think, with pretty a pretty broad consensus that things like second trimester infections increase the risk for schizophrenia. So you don't need the autism story in order to, I think, demonstrate the importance of the work that you're doing. And we might have an argument about, which I'm afraid we don't have time to have here, about your read of the epidemiological literature. I just say it's an area right now that really continues to be extremely hot, particularly in the context of a presidential campaign that includes people that are driving, using a similar narrative in a different way.
No, I appreciate that. Actually, one of the things that made it in my notes but didn't actually make it into my talk was this caveat of epidemiologic studies. And I actually very much agree that there are many limitations, especially with these epidemiologic studies around neurodevelopmental disorders, by and large, not just limited to autism. Right. And so we do have to look with a very cautious eye in the interpretation of that data. I think that the other thing is, the other thing that I think about a lot is kind of chicken or the egg. There are some neurodevelopmental disorders that where kids are more prone to just infections. And so the question is, are they getting infections because they just happen to be more prone? And I think that is actually also a very valid read of the data as well. And so you're very right in that we need to give kind of these counterpoints and caveats here. And actually, this is something that Anna and I talked about yesterday. I just forgot to mention it. But yeah.